Hi everyone, it's Mrs. Piccolini here, and today we're going to talk about a dihybrid, which is similar to a monohybrid, but a monohybrid is where we only look at one trait, and a dihybrid, it's a little more realistic. We're going to look at two traits at a time. And so we're going to talk about that in terms of tiger breeding, which is something that's sort of popular right now, and tiger breeding in captivity and what that can mean in terms of the genetics. And so I had you read an article about white tigers, and what happens with them is that because it's such a rare trait, a lot of times they're inbred, and the trait for that white coat is recessive, but also a lot of deformities such as their eyes are also recessive. And so here you can see the difference between an inbred white tiger and a regular tiger that has a little bit more genetic diversity. And so in order to do the dihybrid, we have a word problem here, and it says the trait to be a white tiger is recessive and having normal eyes is dominant, while eye deformities are recessive. If both parents are white tigers that have heterozygous normal eyes, when they are bred together, what are the chances that the offspring will be white tigers? So again, what they're trying to do is breed the two white tigers together. And here you wouldn't know that they have any eye deformities because they're heterozygous, which means they have a dominant trait. So what are their cubs going to look like? So here, when we do our givens, this is why I had you do things like this, because we're going to have a lot of givens here. So the original trait that we're looking at is a white tiger. So a normal tiger would be a capital A, so that would give you that orange pigment, but a lowercase a would give you that white tiger. It's not going to make that orange pigment. The other trait we're looking at is the eyes. So capital E would be normal eyes, and lowercase e would be eye deformity. Okay. And so now, just like before, we have to figure out our parent cross, but we're going to have to do something a little bit different. So we know that both parents are white. They don't make pigment, so they're going to have two recessives for their coat pattern. And for their eyes, they're both heterozygous. So you put each parent with both traits for each parent. So let's say this is the mom tiger, and this is the dad tiger. All right, and we figure out when we breed these two together, what sort of offspring are they going to make? What most kids want to do, let me erase this. What most kids want to do is just take each one of these letters and put it across this box. So as you can see, this is a 16-box Punnett square, and it would make sense. You just put each letter in one of these boxes. That's actually not how you do it. You actually have to figure out how the combinations would go together. So I like to think of it as foil. So you do like the first A with the first E, the outer A with the outer E, the inner A with the inner E, and the last A with the last E. However you figure it out, each one of these A's has to match up with each one of these E's. So the first A with the first E would give you lowercase a capital E, the outer A with the outer E, the inner A with the inner E, and the last A with the last E. Okay. Turns out dad has the same traits, again, most likely due to inbreeding. So this isn't always going to be the case, but we're going to get the same combinations for his sperm. So now what you do is you put two letters in each one of these boxes. So you put big A, little a, little e, little a, big E, little a, little e. And let's say we put dad's sperm combinations going down this side. So now, just like before, we're going to match the letters so that A from this box will match with the A from this box, and the E from this box will match with the E from this box. And we're going to continue along. So I know I like to put capitals first. You don't put the capital E in front because you still want to keep the E's together. But I do put the capital E in front of the lowercase e, and we go ahead and we fill this out. Okay? If you look at these combinations, all of these tigers are going to be white, but they're all going to have normal eyes because they all at least have one capital E. Okay. So if we continue, we get to this box where it actually has all recessives. So it's going to have that white coat, but now it's going to have the eye deformity. Okay. So as you continue to fill this out, it's going to look like this. And what I did was, when I did my genotypes and phenotypes, was I color-coded it so that it'd be easier for me to keep track. I also like to do fractions here since, you know, there's a lot more boxes, so it's easier maybe than doing percentages. So I do, my options are 2 homozygous recessive A's and 2 homozygous dominant E's. So little a, big A, big E. And there's 4 of those out of 16. I have little a, big E, little E. I have 8 of those. And then I have 4 that are homozygous recessive for both traits. So what is that going to look like in these cubs? 75% of them, or 12 out of 16, are going to be white with normal eyes. And 4 out of 16, or 25%, are going to be white with that eye deformity. So again, this sort of explains the genetics behind breeding and why breeders really have to be educated in their biology and their genetics, whether it's dog breeding or any sort of, you know, any sort of life that you're creating, understanding how this can affect, you know, those organisms. Now I'm going to have you guys try this and some other problems on your own.